I guess we've talked a little bit about variation that exists in the human genome. So what I have here is just a typical genome versus the reference genome. So each personal genome combines inherited alleles and also new variation. And then you've also got your de novo mutations as well. And so on average, this differs from the reference human genome at approximately 4 to 5 million sites. Over 99% of these variants consist of single nucleotide polymorphisms and short indels. So this is based on the 1,000 genomes phase 3 data set. You have 150 to 180 sites with protein truncating variants. So this could be, for example, an early stop codon in a protein coding region. You'll have 10,000 to 12,000 sites with peptide sequence altering variants. So this is in the exome region of the genome. And then you'll have 460,000 to 565,000 variant sites overlapping with known regulatory regions. So this could be untranslated regions, so UTRs. This could be promoters, things called insulators, enhancers, and transcription factor binding sites. So these are in the intronic or regulatory regions. And then you'll have 2,000 variants per genome associated with complex traits, so things that have been picked up in GWAS. And so obviously these numbers can change, and typically your copy number variants have a rate of mutation which is much higher than what you would see in a single nucleotide, for single nucleotide variants. So this is pretty much a table of that information with a little bit more detail. So this is a published paper. This looks at the average sites in the different populations. So you've got African, American, I think this is South American, mixture of American populations, East Asians, Europeans. I'm not sure what SAS is. South Asian, maybe. So here you've got the mean coverage, the number of samples in each population, the variable sites, and whether these are singletons or not. So you've got SNPs, so that's can vary across populations. You've got the number of indels, so insertion deletions. Large deletions are also approximately 1,000, and you can keep going down. And for example, genome-wide associated SNPs, each of these populations has about, each of these individuals in these populations has about 2,000.
Is this from sequencing data or from genome-wide genotyping?
Deeply genome-wide. When you say genome-wide, not SNP arrays. This is 1,000 genomes.
But this is sequencing?
Yep.
This is the entire genome?
Yes. Yep.
So there's only 4 million
sites that differ from the reference genome.
An individual, that's interesting. I thought the number of SNPs was much higher than that.
I think this is variable sites, not total number of sites. So I think total number would be, isn't it, close to 3 billion or something?
3 billion, right. No, but I thought the number I'd read somewhere that's on the NIH website is like 60 million SNPs that kind of vary.
Okay, I'll look that up.
It could be that they're using a different allele frequency threshold for what they're defining as a SNP, but yeah, I'd be curious.
Yes, that's possible.
How can people have large deletions?
Just be something different. On average, how many deviations a given person has from, the reference genome, which is different from how many SNPs there are.
Yep, and that can also, yeah.
How can people survive large deletions? I mean, I guess it's a matter of how large is large, but.
Yeah, there's lots of questions. I mean, obviously, we don't see the people that have those large deletions and don't survive. So, but yeah, deletions can exist in the, we have 2 copies of every genome. There's a lot of compensatory things that can go on. You know, we can have an extra whole chromosome and still be okay. So yeah, I mean, basically it's a complex system, I think, and there's a lot of variation. So, so I'll talk a little bit more about structural variants. So I guess 99% of the sites are SNPs and indels, but structural variants actually affect more bases in the genome. So the typical genome contains 2,000 to 2,500 structural variants, and this includes 1,000 large deletions, 160 copy number variants, a number of other insertions, maybe 10 inversions, and this overall affects 20 million bases of sequence. So part of this includes STRs or microsatellites, so these are short tandem repeats, and also expansions. And I'll talk a little bit about how expansions relate to neurological disorders. So what are STRs or short tandem repeats? So they're also called microsatellites. Basically, these are 2 to 6 nucleotides in length, and these are repeated hundreds of times. So if the DNA sequence of a gene has in the order of a dozen repeats, you don't really see a change in function, but larger areas of repeats can expand during DNA replication, and this is due to slippage events. And the repetitive structure can create unusual secondary DNA conformations that are prone to what I just mentioned, replication slippage, and this results in a high variability in the number of repeat elements over generations, but also in your somatic cells.
What are somatic cells?
So during mitosis, just growth and repair. So all of your cells are somatic, and then you've got germ cells. So the spontaneous mutation rate of these short tandem repeats exceeds that for any type of known human genetic variation. So this can reach 1 per 500 mutations per locus per generation. Yes?
Something about something Dan said. So there is an allele frequency threshold to be considered a SNP, or so basically you have a whole bunch of genetic material. It's not really very frequent minor alleles for that material, and so we don't think of those as SNPs, but for pieces of genome that do vary at a certain frequency, those are what we think of as SNPs.
Yes. So a typical threshold would be something like 1%. If it varies at a 1% rate, is considered a SNP, and anything less than that is considered an ultra rare variant or a mutation potentially. So the issue is, if it's rare enough, then it may have arisen. It may be de novo mutation that just arose in that individual.
I mean, the acronym, the way I understand it, it's SNP, so it's single nucleotide polymorphism. And so when you talk about a polymorphism, then you're talking about something common. So you can define common differently. So there could be 1% minor allele frequency, it could be 5% minor allele frequency. And then you can also term that in terms of SNV. So that means single nucleotide variant. So that could be both either common or rare. So okay, so this is just an example of expansions and their association with many different neurological disorders. So these expansion repeat disorders are considered causal in over 20 different conditions. So there's a list here. And these are the actual causal repeat genes and where they're located. So these include things like myotonic dystrophy, Huntington's disease, and Fragile X. And so the way this is described is you've got a CTG and then you've got it a certain number of times repeated in a particular gene, and this could be in the intronic region. So this is actually the frequency that this occurs is 1 in 8,000 individuals. For Huntington's, it's 5 in 100,000, and for Fragile X, it's 2 in 10,000. So we've spoken about the inheritance pattern of these types of disorders. So typically, it's autosomal dominant with anticipation. So with each generation, you get an earlier onset. You might get more severe symptoms with a greater numbers of repeats. A lot of these disorders also, the expansion size relates to the disease spectrum. So this can contribute to disease liability. And also, as an example, our group has been looking at motor neuron disease or amyotrophic lateral sclerosis. And there's also reports in the literature that intermediate repeats in a gene called ataxin 2 can also increase the risk of this disease. So they're not necessarily causal, but they do change the risk. So, and then larger expansions in this same disease cause a slightly different disorder called spinocerebellar ataxia. So I wanted to talk about expansions because it's an example about how the sequencing technologies don't necessarily do a good job, and still we're not very good at detecting the size of these variants. There's a expansion, again I'm coming back to ALS because that's what I'm familiar with, but in the gene called C9orf72, and this is the most common variant that's detected, but accounting for genetic variation still remains difficult, and this includes variability in the measurement and also mosaicism. So this is differences between cells in actually the size of this expansion. So how do you measure this? How do you pick it up? You can't use SNP arrays, you can't use there is one published method for a next generation sequencing using whole genomes, but that's also difficult because of the short fragmented reads. The way people have been defining or measuring this is that they've been using a thing called repeat prime PCR. So basically instead of 2 primers, you have 3 primers. And this can detect whether you're a carrier or not, but it can't actually detect how big your expansion is. The way you can detect, so this is just a picture of a repeat prime PCR. These are 2 carriers that's generated here, and this is a non-carrier. What is commonly used is a southern blot, but this is an extremely long process. So it can take at least 1 week for one individual. The DNA input needs to be high quality. You need to put a lot in. And it can estimate size, but it's often a guess because you do get this variation within tissues. And I don't know if this is even projecting very well, but I mean, this is how you have to read a southern blot, but you have this is DNA size, and then the lanes that have an X in them are the ones that are the carriers. And you can see this faint line up here, which actually is how you estimate the size of the expansion. And this in a graphical format comparing different tissues. So this is frontal, cerebellum, and blood. And this is the range in the repeat sizes that you might get between individuals across tissues. So yeah, this is just an example of variation that's still hard. We know that it exists, but it's still hard to pick up. And I think the long read technology will be very useful for these types of neurological disorders, but also how might differences in repeat size contribute to complex traits? We're not sure. Yep.
I was wondering, why do these Why do you get different ranges in different tissues?
So during mitosis, your DNA is replicated. And these repeat processes, basically it's hard for the DNA replication machinery to deal with lots of repeat regions. It tends to slip, and during that slippage process, the DNA actually changes and you get longer than expected, and it can create weird structures in the DNA, is the way I understand it. There's some videos that you can look up if you want to see that process. But yeah, it's generally thought as a slippage event during the replication. Yep.
So I was thinking back towards a claim that Dan made the other day about how one of the reasons that we think that genes are so nice to study is because they're fixed at conception. But it sounds like these types of variants are not fixed at conception. And so then we need to worry about I have two questions. First, is there any understanding of kind of environmental types of things that make these slippage events more common? And are there other types of genetic variants kind of similar to these where we might expect a person's genotype to be changing over time?
So to the first question, environmental impacts on these events, I'd say I've never seen any evidence to explain these differences. And for other types of variants, anything I think that has repeats, like those short tandem STRs, we know that they differ between individuals, and I don't know how much they differ within an individual. So, but yeah, I mean, whether this overall, this could be noise. When you're talking about between tissue differences, it could be noise, and you might be interested in an overall effect. So you might not be concerned with differences, but this is just a demonstration to show that there are some differences, but in the whole scheme of things, whether they really impact on what you're trying to measure or what you're trying to understand, it depends on your question.
Thank you.
Yeah. Yes.
Along those lines, so are these expansions, they're not normally picked up on the genotyping arrays? Is that what you said? Or
Yep, they're not tagged by SNPs.
And things like that. So.
Well, the ones I know about, they're not tagged by SNPs.
Okay. So it wouldn't necessarily be something.
There's certainly risk alleles that are associated, but they're specific, but they're not very accurate in that you can define a proportion of, so you can say everyone's who's homozygote for this or heterozygote for this, who carries this allele, is more likely to have this carrier expansion. But then of that proportion of so many individuals, that the proportion that has the expansion is much smaller than the people that actually carry the risk allele. So
if you think of changes in the somatic cells, cancer is a big one. And is
cancer.
Cancer mostly
Good
about
point.
SNPs, or does it have these kind of things too?
Cancer certainly has these types of things because, you'll get a mutation, a somatic mutation, and that can cause the cancer. And then you'll get this excessive growth in these cells, and then these will accumulate mutations.
No, but I mean, in the somatic mutation,
yes.
is cancer often copy repeats, or is it mostly, maybe SNP is the wrong name for it, but sort of a change at one base? Would change at individual bases cause a lot of cancer in?
I'm not sure about the
somatic
genetic
cells?
architecture of cancer causes. Yeah, I would suggest that they could be either. Yeah. Okay, so I guess we spoke about technology, and so when you're deciding on a technology, it really depends on a number of variables. Of course, WES or whole exome sequencing might be better at looking for Mendelian diseases, as mutations in protein coding regions we know are enriched, but only account for 11% of GWAS hits. SNP array, of course, is very good at tagging the entire genome and is likely better for complex traits and diseases. It's a very efficient sequencing, particularly for large populations, which we know we need in these genetic studies, looking at these complex traits. 57% of non-coding GWAS hits lie in broadly defined DNA's hypersensitivity sites, and this consists of 42% of the genome. So what I was trying to bring about is that you still need to be aware of, maybe even account for if you can, mutations in non-coding regions, as these can also cause Mendelian disease and potentially influence human variation and complex diseases. The integration of whole genome sequencing, long reads, efficient and cost-effective pipelines over the coming years will certainly would be the ideal choice. This is just a summary table for your reference of the different ways you can look at the DNA, what the outcome measures of these are, and again, what are the challenges to interpreting each of these data sets. And as you know, for a genome-wide SNP array, often the top SNP or identifying the causal variant isn't necessarily the causal gene, and we'll talk about that a little bit later. There's overall, I think we're quite aware that there is a very large amount of diversity in the human genome. Not all the technology accounts for this perfectly, and there is expression data, functional models and systems that can complement and help direct investigations, and that's what we'll talk about next. So the summary of what I've talked about so far is population genetics. Common alleles are shared between populations. Genetic variants on the same haplotype are linked. A large fraction of alleles have low allele frequencies, and genotypes and alleles are in equilibrium in an infinitely large population. So that refers to the Hardy-Weinberg equilibrium. Genotyping technologies, differences in accuracy, efficiency, throughput. There's a large amount of diversity, and structural variation and expansions can be highly variable between individuals, and the variation is not necessarily accounted for in genetic studies. So for this next two lectures, I'm going to be talking after lunch as well, so I'll see how I go. We'll talk about how genetic variation is described, so just the general nomenclature, and then we'll talk about functional models. I'll give you some overviews of what animal models are currently being used and have been used, and how these systems can be useful, and then how do you interpret and apply this to human genetics. Okay, so how is genetic variation described? So this is just a very basic slide, and some of you might be familiar with this already. So the reference genomes and genetic scriptures have been changing over the years. Essentially, you need to describe the DNA change with the relevant RNA sequence using neutral terms. So typically you say variant and change rather than mutation. And so this is how you might see something written in a paper. So this refers to the reference sequence, and then this is C, meaning this is in the DNA, this is the base, and then it's a change from a C to a T, so a cytosine, to a thymine. So the NM is just the reference sequence based on the RNA, and I should say this is actually the version. So this gets updated even quite regularly. So if you you know, it could be different one day to the next day because this reference sequence might get updated. And
So that's just like a particular person, like it's just one genome that we're deciding is the reference sequence? Or
yeah, so this is a reference mRNA, and then you want to say what the change that you've identified. And so depending on the reference sequence and the version can actually change this base number with these annotation updates.
gotcha, yeah.
So a reference sequence that with a version number of 3, this number might actually change. So these are just little things that to be aware of.
Like, I guess my question is sort of like, how do we pick the reference sequence? It's just like, we like started with one and then we'll just use everything relative to that sequence, or is it like the most common one, or?
I think there's a standard reference sequence that everybody uses.
Yeah.
But is it an individual person, or is it like the modal base at that location?
I don't know how it's decided.
But the important thing for us to know is just that everything after the reference sequence is all like contingent on the reference sequence.
Yeah, that's right.
Cool.
And the version number.
Cool. Thank you.
Yeah. So I've just described this. So the C nucleotide at this particular position is changed to a T. There's also RS numbers. So these are, this RS is a reference sequence. And you'll see that other transcripts exist under these names. So this is how you would describe the protein change. So this is the particular gene. This is the protein sequence, the version number, P meaning protein. So it's a glycine to a histone. And that's the amino acid position that's been changed. Oh, it's a glutamine, sorry. NR, so NR is just a reference sequence based on non-coding RNA. So if you see NR, then this is not mRNA, it's actually non-coding RNA. And the chromosome position depends on the build and the assembly. So yeah, that's just an example of, usually it's HD 19 or HD 38, so this can depend on the position. What else was I going to say? Okay, so these can be condensed into descriptors, so whether something is loss of function, for example, a single nucleotide variant that are frame shift, or a stop loss, stop gain, a start loss. You could have a splicing donor, acceptor, and frame shift indels. And of course, there's non-synonymous mutations. So these are things that don't actually change an amino acid. Non-synonymous does change an amino acid. So missense single nucleotide variants and non-frame shift indels. Okay, so there's scoring methods that actually exist for categorizing and predicting variant impacts. So some of these methods include polyphen, SIFT, CAD. There's also a few others now. I'll just provide you with an example of CAD. So this stands for Combined Annotation Dependent Depletion. So it's a measure from the Schenger lab at the University of Washington, and this basically summarizes evidence from 63 different annotations, which encompasses functional or regulatory annotation, allele frequency and diversity, and also evolution conservation. So they're drawing from a lot of different pieces of information to understand the impact of a functional variant. So it attempts to give you an unbiased prediction of deleteriousness, and this is based on a machine learning comparison of 15 million observed variants and also simulated human variants. So they provide a raw C-score and a Fred-like scaled score, and this is normalized. And they say both scores are useful depending on what your question is. To provide you with an example in terms of the scale score, if it's scoring 30 or above, so this is the x-axis here, then it's the top 0.1% of likely deleterious variants. 20 and above is the top 1%, and then 10 is the top 10%. So these are enriched for things like stop loss or stop gain. So these are predicted to be more likely deleterious to function. So this can be useful in understanding the variant impact, particularly in the coding regions of the genome. And it's also been used in GWAS as well. So, but you know, this is still just a prediction. So it's a useful resource. It is improving, and there's a lot of different scoring mechanisms coming out. But again, they are just predictions. So I'll talk about the use of functional models. So at what stage would you be interested in a functional model? When did they become useful? So you might have loci or variants that are identified in your disease or your trait of interest that meet your genome-wide significance thresholds. Then you might have independent replication of these loci, giving you more confidence in that initial discovery result. And then you might find that your variant or loci is functionally annotated in those available tools and databases, and you know, is predicted to have play a role in what you've identified is an area of interest in the genome. So from there, ideally what you want to go on to do is some type of experimental validation or a model system to look at the gene level function. And the other thing is you might want to use a functional model to actually narrow down your region. So often you might get a SNP hit in your GWAS, okay, it's replicated, but in actual fact, the SNP might be in linkage disequilibrium with a number of other SNPs. And of course, we know empirically that the gene that is the closest to the SNP, even if this is not necessarily the causal gene involved in the trait or disease of interest. So these types of models can help you understand which gene is causal. Okay, so animal models. So these aren't recent. They've been used for many, many years to understand the genetic influence related to phenotypic variants. And they've actually been used without knowing the genes of action, but they have used to model theory of genotypic and environmental components. So there's been a lot of breeding for selection. So examples being you can select high or low lines. So this could be based on activity, for example. And you can continuously breed those over 30 generations and continue to increase your trait. And the way this works is that the difference is increased slowly each generation, and this means that many genes are contributing to the variation in the behaviour. And of course, if the trait was a simple Mendelian, i.e. controlled by a single locus or a single gene, then the lines would separate after a few generations and wouldn't diverge any further. If intensive inbreeding results in almost an identical genetic background, and so this can be used to model single variant changes or environmental impacts. So you'll be comparing littermates, and then you'll have a single gene that has been knocked out or overexpressed, and that gives you smaller variants related to genetic background. So you can just narrow down on what the effect of that single gene is. So that's the benefit in that particular approach. There's also been comparison, so there's quite a few different inbred strains that exist, or genetic backgrounds. And these genetic backgrounds have actually been compared. And so I'm particularly talking about mice here. So they've produced a catalogue of differences that have differences in anxiety, learning, memory, and stress reactivity. And this is available in the Phenome JAX database. Yes.
Suppose you took GWAS results and you had this polygenic score for something like educational attainment. Now,
Yeah.
could you, with a huge amount of work, let's say, okay, here are the corresponding SNPs in these mice, and so let's measure the corresponding polygenic score as well as we can in the mice, and then see if those mice were different. So can you translate across species a polygenic score with some degradation?
I haven't seen that done, but yeah, I'm not really sure how you would do that.
I mean, can you kind of, I guess one way to ask the question is how many sort of
So what
chunks
mice
of
would you
DNA
do?
would you know what the corresponding chunk of DNA in the mice was? Are you typically able to say, okay, here's the chunk of DNA in humans, here's the corresponding one in mice, or this is one that's only in humans, maybe you leave that out? How many could you map over to mice?
I think the answer is most.
Yeah, I think that yeah, mice are very genetically similar to humans. 97%, 98%. But in terms of getting the polygenic score being for a particular mouse being exactly what it is in a human.
No, it wouldn't be exactly, but the question is how much degradation?
Well, there's two issues. One is how much of the genome is shared between mice and humans, which is very high, and the other is how many SNPs are shared. Because a lot of the SNPs that vary among humans don't vary among mice and vice versa. So, you know, I asked this question to, I've asked this question, similar question to several people who work with animal models, and what they've, you know, I think they all think it's an interesting idea, and as far as I know, no one is trying it, and I don't think we really have a good sense of how far you could go with it, and
Yeah.
Seems hard to make universities for mice too, so like, I don't know.
No, but that's the question, is what shows up differently in mice. I guess you'd probably have to assume you'd need full genome sequencing to do this.
Yeah, I don't know whether that
You wouldn't
exists
have
in
a
all the
linkage
strains.
disequilibrium, anything like for humans.
yeah, that's another issue. So the linkage disequilibrium, you know, unless you know, since you don't know the causal variance, and the linkage disequilibrium is going to be totally different, you can see a lot of things much worse than going to African ancestry populations.
You can do something sort of gene-based or functional-based? Is
Yeah,
that
potentially. I mean, I think the idea has
So
some.
rather than the SNPs really
Yeah.
detect a read, okay.
It's just a matter, you know, there's just a lot of hurdles to be overcome, but it sounds like there's some.
Yeah, if you have a huge number of full genome sequencing for humans, then that's one of the biggest hurdles to get over.
So James, say something.
So my take is that it's already clear that there's a lot of use that can be gotten out of results in mice in application to humans, or the other way around even. So, for example, Michael Goddard had a paper a few years ago where he took the GWAS hits in humans. For height at that time, there were roughly 180, or maybe exactly 180. Looked at the closest genes to those SNPs, and most of them have orthologs in mice, or not mice. He was looking at cattle. And then he looked at, gene mapping studies in cattle for body size, and found that there was a strong, above baseline level of overlap, so that genes that affect body size in humans also seem to do so in cattle. Also, this database that the URL is up there, that's also incorporated as part of DEPICT, which is the gene set enrichment tool that you can apply to human GWAS. And actually, very often, this is one of the data sources that provides the best results, so to speak. So, for example, if you take these genes that are annotated as causing these certain phenotypes in mice in these knockout experiments, so if you look at height, the top results include like the decreased embryo size. That is, genes that if you knock them out, one of the resulting phenotypes is that the mice can embryos are born smaller, or such genes also tend to be near SNPs that are among the top results that actually ones. There was a certain point, this has changed as more data, decreased brain size, which is a mouse phenome category. If you knock out these genes in mice, one of the resulting phenotypes is a reduction of brain size in mice. There was a certain point in time when this was the #1 most significantly enriched gene set according to the PIC for years of education. So clearly the animal data and the human data reinforce each other very well at this point.
There's one other point about these animal models is that because, as Fleur is saying, that they're carefully controlled environments and are often interbred to have very similar genetic backgrounds, so you minimize the noise that comes from epistasis and things like that. The effect sizes for in terms of variants explained for individual genetic variants in animals tend to be much larger than the corresponding effect sizes in humans, which is why these animal studies can be done typically in much smaller samples.
Yeah, that's right.
Oh, there's a caveat to that. So a few years ago in Nature Reviews Genetics, Michael Garton did a paper where he sort of talked about his experience going from animal genetics to human genetics, and now is occasionally going back. And he basically conceded, yeah, I realize now that when we were just doing livestock genetics, we were kind of doing things kind of wrong. That we didn't use stringent enough significant thresholds or large enough sample sizes. And our experience in human genetics has wisened us up. So, there's that.
Okay, so basic research using animal models is a feasible way to establish causal relationships. And in the reciprocal interactions among social, behavioural and genetic contributions to health and disease. So there's natural occurring variations as well as mutation creation, and this has been done in multiple different models, so not just mice but also things like bacteria, C. elegans, which I'll talk about, Drosophila, zebrafish and mice. And so the field, I think in all of these respective animal models, is moving into a more high throughput approach and multi-gene models to really characterise function. So I'll just talk through each model one by one. So C. elegans, also known as the roundworm. So it's actually extremely small. It's only 1 millimetre in length. It has a very short lifespan, 3 weeks. And the great thing about it is that there's only 959 cells in this organism, and 300 of those are nerve cells. It's a very anatomically simple and extremely well characterised nervous system, and has helped contribute to understanding of the neuronal system, how it actually works in an organism. It does actually include a primitive brain, which is a nerve ring, and they have identified variants that alter foraging, memory and learning. It was actually the first animal that was sequenced in 1999, and about 50% of genes have a human ortholog. There was a review in 2005 that mentioned that it's been difficult to connect the dots between genes, brain and behaviour in this particular model. They do have a behaviour database, and this was created for rapid and consistent quantitation of these behavioural phenotypes that they're measuring in these worms. So another model that exists and has been widely used is the fruit fly, or the Drosophila. So it's slightly larger, 2 to 4 millimetres in size. It also has a short lifespan, and it's also there's a high productivity, so it can produce 500 eggs. And there is a process for simple gene manipulation. So it's been studied to look at mosaicism and also the X chromosome. It has a more complex nervous system, so it's got neurons and glia. It's protected by a blood-brain barrier, and there's organisational similarities with the vertebrate brain. So it's not exactly the same, but there are similarities. There's a thing called Flybase. So this is an online database which is dedicated to Drosophila. They've got 14,000 genes in there with tracks on all available data. So the link is there. About 75% of known human genes have a recognisable match in the genome of the fruit flies. There's transgenic flies, so these are a collection of fly lines, each carrying an inducible RNA construct against a single protein coding gene. So these are essentially knockdown fly lines. And so there's also a link there to look at that. This is just an example about the characterization that you can actually do in a fly. This is modelling a human neurodegenerative disease. You can look at retinal degeneration, so you can look at the control and the disease model. They find that there's a pseudopupil loss. They can measure neural degeneration in the model compared to a control fly. They can look at the brain vacuolization. So this is the control versus the disease. They can look at neuromuscular junctions. I think the neuromuscular junctions do differ, but of course it's still, you can look at the change in its structure. You can do a lifespan analysis between control and disease, and also there's different functional assays, and here they've done a climbing assay to look at functions. So there is, although it is just a fly, I think there are still some important assays that you can look at with much greater depth than you could do in a human, for example.
What is mosaicism?
So different cells having a different genetic variant. So
how did the different cells get a different genetic variant?
So like a somatic mutation, for example. Also, there's X inactivation. We know that occurs very early on, and then all daughter cells that are coming from that cell division all have the same X being inactivated, so that's also an example of mosaicism. Okay, so zebrafish, I guess this is maybe a more recent animal that's begun to be more widely used in the literature. It's about 4 centimetres long, has a 5-year lifespan, again, has quite high productivity. It's been a key vertebrate for actually studying the development, as it can be observed, the embryo can be observed directly, as it's not within the mother. So here you can see just the development of the embryo into the fish. Over 2,000 genes have been identified that affect this embryo development, which is quite interesting. And recent models include both sensory and motor development, social behaviour, and associative learning. And actually a very good paper that Jian Yang in our lab often brings up as a great example of how you can use these animal models is this Golzio et al. So it was published in 2012 in Nature, and what they did is they investigated a section on chromosome 16, which was a region that was really frequently deleted or observed to be duplicated in psychiatric disorders. And so they actually narrowed the effects on the brain to one particular gene in that region. And the way they did that was they modelled each one via knockout and overexpression. They also did some pairwise overexpression experiments and found that there were 2 other genes that also interact with the gene they thought was having the largest effect. And that found that it could actually increase its influence on brain size. So I guess this is just a good example of how a region was narrowed down to particular genes, and then you could understand, from there you can go on to understand more biological pathways or mechanisms that are going on once you narrow down to those genes. You can also perform gene silencing experiments in the zebrafish, so you can use double-stranded RNA to knock down genes. So this is RNA interference or small interfering RNAs, and this is most successful in cells and simple models, and also applies to the C. elegans and Drosophila.
What do you mean by knockdown genes?
Simply that, just knock down the expression.
Knock down the expression of the gene. Okay.
Yep.
So that's different from knockout.
Yes, different mechanism to, knockdown is the gene is still there in the genome, but you're using RNA to knock it down, but knockout, you're changing the genome. Yeah,
So why
the
would
way
you
I
do,
interpret.
when would you do knockdown and when would you do knockout?
I think knockdown is actually easier, so it's more like a high throughput type approach.
Knockout stuff wasn't possible until CRISPR was invented in the past couple of years. So not easily possible.
I think knockout existed before CRISPR, but it was harder. So you take many more months to create your chimera and then breed your homozygote knockout. So yeah, there were methods before CRISPR. CRISPR has improved the efficiency of it. And these RNA approaches are also more efficient, particularly for these particular animal models, where you can breed many animals in a very short period of time.
Can you knock them down even during embryological development?
Yes, and that's how they've discovered that over 2,000 genes impact the embryo's development. Okay, so rodents, mice and rats, so these have been a key mammal for mutational screening. The mouse genome was sequenced in 2001. Nearly all genes are similar to humans. The chromosome is also in a very similar order to humans. I guess relevant to the behavioural field of research, I think rat is generally the preferred model for measuring physiological and behavioural traits, but the problem is that knockouts have only recently been able to be efficiently generated. So I think they had a lot of problems culturing these the rat cells, which they didn't have the same problems with mouse cells. So the great thing about this model is that you can do targeted mutations. So over 10,000 knockout mouse lines have been created. It's a good system model. So, it's a complex system that, has a brain, has a neurological system that's more closely related to the human than it is, for example, in the C. elegans. If you knock out a gene from birth, we know that it results in compensatory changes, so these aren't necessarily a direct function of the gene. And so now there's ways that you can do conditional knockouts, which you can turn expression on and off throughout life, or in specific areas. So you can do conditional knockouts specific to a particular tissue. There's also been mapping of the quantitative trait loci. So this is mapping selectively bred lines and to identify regions that are contributing to, for example, a drug response or activity. And often, however, these are too large to actually pinpoint single genes. But you can also do gene expression studies to kind of understand the pathways or mechanisms that are leading to this differences in, for example, activity. So this is just an example of this selective breeding. So you have outbred lines, you've got high and low, and so over these successive generations, almost 40, you can see that the running distance of this, so this is in rats, does increase with each generation. Of course, you might want to look at the pathways that are associated with this change in function that is hugely different from the low selected lines. This is just an example of what you might see when you have a knockout. Here is your wild type and a particular gene. This is a protein blot, so you can, this is using tissue and then using an antibody to detect whether the protein is present. For these 3 animals, you see the protein here, and for the knockouts, there is no protein. And of course, you can pick up compensatory effects. So this was the mouse model I used in my PhD, and basically what we found is that when we knocked out a fast twitch muscle fiber protein, what we got is an upregulation of the muscle protein that was highly related but typically expressed in the slow muscle fibers. So I think you can use these models well, but as long as you recognize the differences between the human and mice. And so, for example, when we had this in the mice, we were interested in muscle function, and we're very well aware that mouse muscles have a lot more fast fibers than humans. So humans have a fast fiber proportion of maybe 10%, maybe 15%, and in mice the proportion was much larger, 80%. So we knew that any effect that we would be generating by knocking out a fast twitch muscle fiber protein, we knew would be enhanced in our mouse model relative to our human model because of the differences in fiber type proportions. So, these are unique system differences that you just need to be aware of when you're modeling something in a different organism. And the same with neurological considerations. So I think in the fly doesn't, in the neuron isn't actually myelinated. So if you were looking, so I know Charcot-Marie tooth, a lot of those, the variants associated with that disease impacts myelination. So this is the sheath that covers the neuron that helps conduct the electrical signal. Maybe it's not the best model to use a fly if you're interested in effects on a myelin sheath. So yeah, just being innately aware of the system and its limitations, I think, is important. So the great advantage of these animal models is that you've got this intact system to model a genetic effect, and you can do this in various ways. You can look at not only genetic causes, but also genetic modifiers. I've just put a picture here of a genetic modifier. I mentioned ataxin 2 as being associated with neurodegenerative disorders. There's a causal mutation in TDP, a gene called TDP43, and then when you include variants in ataxin 2, this actually makes the disease model worse. So it kind of correlates with the human genetic findings that we're finding this increased risk when you have this slightly larger number of ataxin 2 repeats. The other great thing about animal models is that you can use and examine whole tissues, whole organs at different time points with different exposures. And that's just something that you can't do in humans. And you can also control for confounders. So as Dan was saying, you can collect animals at a similar age. You can control the rearing, you can control their diets or other environmental concerns, or you can look at the effect of the environment. So there's various interventions that you can do to actually understand an effect. So limitations, they're not humans. So, and we know that species-specific differences do exist, and you really should consider this carefully when you're interpreting your results and developing your animal model. So if the function of a protein or expression of a gene has changed, then you should recognize this when you decide on the model, as I've mentioned. I should talk about, of course, animal welfare. The way these studies work is that you should always endeavour to maintain 3 R principles. So the R principles are reduction. So when you perform a method, you should try and do it from fewer animals, or aim to obtain more information from the same number of animals. Refinement, so this is use of methods that alleviate or minimise potential pain, suffering or distress and enhance animal welfare for the animals used. And replacement, so preferentially use methods which avoid or replace the use of animals in research. So yeah, obviously you don't want to use them if you don't have to. And I think there's some exciting humanized models that could be more relevant or more useful because they're closer related to humans than these systems. So I'll talk about cell models. So cells can be somatic or stem cells. So the difference between the two is that stem cells have a capacity to self-renew. So there are a few limitations of somatic cell lines. So this is generally somatic cell lines are those that are associated with a particular tissue. However, some of the more high throughput analyses of these lines have suggested they don't necessarily represent the tissue. So looking at the expression, they've shown these tissue-enriched genes are actually downregulated or completely turned off in the corresponding cell lines. In 1998, there was a paper talking about the issues with false cell lines. And then a paper later in 2007, almost 10 years later, said that it still remained a problem. So 18 to 36% of cell lines were incorrectly designated, yet continue to be used and circulated. So this is also something that you should be aware of. Excitingly, there's been this development of iPSC. So iPSC the method was first published in 2006. So this is the creation of cells that were actually similar to embryonic stem cells. So these were generated from mouse somatic cells by adding transcription factors to become what they call induced pluripotent stem cells. So pluripotent meaning that they can diverge to any type of cell or tissue. So in 2007, they used human fibroblasts and converted these to iPSC using a similar method, so adding these transcription factors to the cells. So there's a lot of advantages with this. They're human origin, and because they're a cell line, they're expandable, they're accessible. You can produce large quantities of these disease-relevant cells in cell types that have been previously inaccessible. So these are things like neurons, cardiomyocytes. You can look at controls and non-disease affected individuals. And however, iPSCs, they do exhibit line-to-line variations. This picture is actually just demonstrating the uses of iPSC, and CRISPR-Cas9 is there as it's a genome editing technology. So what can this do? Basically, you can create specific gene variations specific to your particular base of interest, for example, and then you can also create an isogenic line. So you can literally just mutate that particular loci or variant of interest, and then you can have an isogenic line to compare that with. So the only thing that differs is a single base change that you've made using this CRISPR technology. So this helps you identify true pathology while avoiding confusion with other disparities in genetic background. And then, of course, you've got the potential to develop personalised medicine, patient-specific iPSCs. So this is just showing you can turn these iPSCs into many different types of cells, so your cells of interest, and of course you can use them for different methods. So I have a slide in here about CRISPR. I'm not sure, I'll just go over it briefly. We've got time, so what does CRISPR do? So CRISPR-Cas9 can actually cut sequencers. So Cas9 is actually the thing that does the cutting. And then you can combine it with a complementary DNA to create specific variants. So this includes the explicit introduction of DNA sequence changes, and you can do this in a precise monoallelic or biallelic manner with very high efficiency. So with the uptake of this method, there's certainly been reports of off-target effects. So this is, you insert your CRISPR-Cas9 and then you're going for one particular area of the genome, and then for some reason something else happens somewhere else downstream. So they have, this isn't all that common. They've developed different versions of this nickase, this Cas9, and these are being used to kind of mitigate this potential effect. But I think this is these off-target effects have more been seen in the cancer field. At the moment, this methodology is very well suited for Mendelian disorders that have an early onset. And I'll show you in the next slide, but when you're working with cells, these are relatively immature. So, and in general, the cells, even though you can, you're creating cardiomyocytes or neural cells, they still exhibit fetal-like properties. So, Mendelian disorders might be good in that you might be expecting a larger effect, and you might be expecting that to happen early on. At the moment, the field, I think, is working to generate isogenic lines that only differ at the risk variants, and of course develop co-cultures. So something that's more representative of a system, because remember, we're doing isolated cell work, and this is different from working in an animal model where you're working in looking at an entire complex system and how single effect happens. This is very narrow and very specific to a cell type that's very immature. So things that are going on in the cell field, I think that they're getting faster and more stable, resulting in higher maturity and purity, and this will obviously benefit for the translational aspects. The great thing about iPSC is drug screen potential, so or as a validation tool as well. But you do need large amounts of cells in a certain population that are sorted and purified before you start to screen these compounds. So you can test for changes in function, but you can also look at toxicity. So organoids is, a step above cells. So this is a 3D culture system. And these have actually been generated for brains, kidneys, liver, and lung, possibly other tissues. And so this is using both tissue stem cells and also pluripotent stem cells from mice and humans. And these are functionally more mature than your typical iPSC. And the limitation at the moment is getting the nutrient supply, I think, and with like a vascularization. But these are being used. I found an example, there was a study in autism. So parent and the proband cells were actually cultured into brain organoids, and they looked at changes in development and expression. And so, yes, this is more mature than a neuron, but they said it was actually equivalent to the first trimester of brain development. So, yeah, I mean, it's quite interesting and a little bit scary that they're creating these brain organoids. But yeah, I mean, it's a rapidly developing field, and these models could be and are more human than going to an animal model system, and are very specific as well, which I think is great for behavioral research.
So I don't know hardly anything about this, but my understanding was that we start with these stem cells, right, and we try to do something to them to induce them to become whatever tissue we're interested in.
So you could add tissue-specific transcription factors, and then you can convert them into your cell.
My understanding though is that we don't actually like. So we create this thing and we think like, oh yeah, this looks a little bit like a neuron, but there isn't like total certainty that what we're like what's produced by these stem cells is a neuron of the same type that we would see in like the developing brain. Like, is it there?
I think there's general, so the way they're working at the moment is there's general markers. So if you test for those tissue-specific markers, then it's generally considered that your cell type is what you are expecting it to be. But yeah, there's a limitation to its maturity. And in general, they're pretty immature. So, yeah. And so I mean, you can look at the maturity of the cell as well. You can test for that by looking at some tissue-specific markers of maturity. So. Okay, so just to summarize what we've just talked about, so modelling a genetic effect in a system remains important to understand cause mechanisms, and is also relevant for therapeutic testing and development. Of course, every model seems at this moment has its advantages and different disadvantages, and the context should be recognised. There's a lot of value in doing it in a complex system, particularly one that's well characterised and mature. And things can be collected and analysed with relative ease. And as I mentioned, this includes things like the C. elegans, the fruit fly, zebrafish, and the rodents. iPSC is now being widely used for disease modelling, drug discovery, and cell therapy development. The cells still remain immature, and there's limitations relevant to system-wide diseases, because obviously you're looking at a single cell type. And also might be limited when you want to measure higher order functions, things like learning, memory, emotion, or late onset diseases. So there's further development is required in this human iPSC technology, particularly with the gene editing and the organoids. And this will make the iPSC-based platforms more powerful in each area, including precision medicine. Humans are always the first choice, I think, and data and resources do exist and are expanded for human expression pathway analyses, and that's what I'll talk about for my last lecture.